So I'm very happy to welcome everybody this evening to our Color Research Society of Canada talk. And tonight we have a very special talk given by our student award winner, Trevor Canham, who's studying at York University with Michael Brown. And I just want to do our land acknowledgement, a little bit of an introduction to the CRSC, and then I'll introduce Trevor. The CRSC's activities related to sharing color knowledge take place across Canada, understood as part of Turtle Island, the ancestral homelands of over 630 First Nation communities, representing more than 50 Indigenous nations and languages. We work to respect, affirm, and support where necessary the inherent treaty rights of all Indigenous peoples across these lands and waters. Our commitment to honoring the knowledge, ways of knowing, and wisdom of Turtle Island's Indigenous peoples aims to contribute to the decolonizing work required today for moving towards a just society. So welcome again. My name is Robin Kingsborough. I'm the president of the CRSC, and I welcome you all this evening or this afternoon, depending on where you are, or later in the evening. We stretch across Canada, which is great. We are a nonprofit organization for color research focused on fostering a cross-disciplinary sharing of color knowledge. We seek to develop and support a national cross-disciplinary network of artists and designers, scholars and practitioners with an interest in engagements with color, and to encourage discourse between art, sciences, and industry related to color research and knowledge. We are the Canadian member of the AIC, the International Color Association, and they're having their midterm meeting this week in Sao Paulo, Brazil. Don't think any CRSC people went down for that, but some of our other sister organizations members are there. I'm very happy this evening to introduce and to welcome Trevor. Trevor was our 2024 CRSC Student Award winner. We started offering student awards for undergraduate and graduate level work a few years ago, and Trevor was our Head and Shoulders winner this year. He received a BSc in Motion Picture Science from the Rochester Institute of Technology, and in the following years, he worked in Marcelo Bertalmio's image processing for enhanced cinematography lab in Barcelona, Spain, and later at the Spanish National Research Council. He's also worked in the cinema industry as an intern at company 3NY and as colorist for several independent films. So I know I'm going to be asking about that too after his talk this evening. In 2023, he was awarded Best Student Paper at the 31st Color and Imaging Conference. His interests lie in the interaction between color phenomenology and imaging systems. So I'm very happy to welcome Trevor and excited to hear about a novel technique for visualizing multispectral images. So take it away, Trevor. Oh, let me see if I have set you up to share screen. Okay, should be all set.
Thanks, Robin. All right, let's get that going. Okay, so can you see my slides? And not my notes?
All good, all good.
Okay, amazing. So thanks, Robin, for the introduction. I'll be presenting Noise Prism, a novel multispectral visualization technique, which is work that I did with Javier Vasquez Corral, David Long, Richard Murray, and Michael Brown. This is a collaboration between York University, Universitat Autónoma de Barcelona, and RIT, the Rochester Institute of Technology. So considering all audiences in the talk, we're going to start with basically an overview of the sort of color representations that we take as the source for this type of technique. then we're going to go into sort of a basic demonstration of the technique itself, followed by an overview of how it works, more or less, what are the technical components, and um, we'll follow that with another demonstration, and then finally there'll be a bit of a discussion on future work. So, without further ado, um, so we're talking about spectral power distributions, or the physical correlate to color perception at its, let's say, highest resolution. Um, we start with an illumination source, which has a particular function with respect to the wavelength of light through the visible spectrum. There's a relative intensity function at each wavelength, which over at the short wavelength, this would correspond to blue colors, and at the longer wavelength, this would correspond to red colors, and then, you know, it's Roy G. Biv backwards across the function there. But we can't just have a light source, or at least we shouldn't look into light sources directly. We want to look at them reflected off of objects, and that's really the idea, is to be able to see, what reflective objects are in a scene, and get to know what's there. So the mathematics for this is actually quite simple to compute the spectral power distribution that's reflecting off of a particular object. You just take this function of intensity over wavelength and this function of reflectance, and for each sample across the spectrum of light, you multiply the two, which is represented by this little uh, point symbol. So that's the physical stimulus. And then there's the interaction with the observer. So the observer has what are known as has a sort of spectral sensitivity, let's say. Um, Trichromatic vision tells us that this comes in three particular bands, which are to some degree overlapping, shown at the bottom of the slide here. And through some clever experiments, we were able to derive what exactly are the ratios for each wavelength in terms of our sensitivity to color. So at this point, um, we no longer are working with a full resolution spectral signal, but rather a tri-stimulus signal, meaning just three numbers integrated over these sensitivity bands, um, which inform us as to what is and what is not a color match. Obviously, there's a lot more to the uh, visual process and uh, the pipeline of processing visual signals, but this work is just going to stop basically at the surface of the visual system um, and address this property of spectral sensitivity. So anyway, we have a physical stimulus, we have an illuminant, it's reflecting off of a reflective object, and it's being observed by some observer. And we represent this with three numbers, which we call X, Y, and Z. The interesting thing about this, and the thing that's lost in reducing the dimensionality from this full spectral stimulus to just three numbers, is that we can have two different spectral power distributions, or SPDs, that trigger the same visual response. And this is actually what allows us to have cameras and displays and things which don't require, an infinitesimal sampling procedure in order to reproduce colors that look similar or are maybe a perfect match to a real world stimulus. And this makes things a lot simpler computationally, so it's kind of convenient. But basically, to this point in our evolution, we haven't needed more than this many channels to specify the difference between, um, you know, real world stimuli that we need to assess to survive. So anyway, there's these two spectral power distributions, and although they're very different in terms of shape, once they're integrated over these sensitivity curves, they end up looking the same to this observer. And this is a concept known as metamerism. So metamerism can happen in two different ways. You can have a reflectance metamer, where you have two different reflectances that under the same illumination source appear the same, but perhaps under a different illumination source appear different, which is in this first set of observers. And you can also have an illumination metamer, where you have two different illumination sources, which, even though the illumination sources are different, to a certain reflectance and to a certain observer, they'll appear the same. So we'll work with illumination metamers in this, with this work. And like I mentioned, we're not going into even the next stages of visual processing in the retina, but rather we're just looking at the effect of the sort of spectral response properties of these pigments, which for humans, we have just three um, different types of these, but as you can see on the right side of the slide here, other animals have more of these, which leads to a sort of intriguing pop science question of what is the visual experience of these other animals that have more types of photopigments with which to differentiate colors? Can they see more colors than us? Are they seeing new colors that we can't even imagine? So this is, something I remember debating with my friends as a kid, and still it's sort of up for debate and something that I have thought about a lot in the process of doing this work. Because the goal of this work is to take metamers, colors that appear similar but have different spectral power distributions, and to figure out a way to represent them continuously throughout an image, for example, such that they can be differentiated one from the other. So enter the noise prism. The noise prism works sort of like a real prism in that it takes the incoming signal, the incoming multispectral signal, and divides it up spatially in terms of its component parts. So you can see now that these two stimuli that appeared the same when integrated over the spectral sensitivity of an observer now look different from each other. And you can tell certain things about the properties of these functions based on the appearance of these patterns. So you could think about it, if you have sort of a mathematical intuition or language, as taking color signals and representing them in a higher dimensional way. Obviously, we're doing a trade-off here. Since we need to spread out the signal, we can't just represent it with one pixel anymore. We lose some spatial resolution, but the goal is to get more color resolution and to engage sort of a higher dimensional visual process to differentiate between these things. And the idea of how these are constructed is basically to think about, if we shrunk those visualizations down, they should sort of integrate spatially into the colors that they were to begin with. And perhaps between these two displays, you can see that those now look quite a bit different, those two pairs of stimuli, or they look a bit more similar, let's say. So this is a part of, or a contribution to the field of multispectral visualization, which to this point has relied on color blending techniques, where they will sort of take multispectral signals and assign three primaries to them, fitting into the traditional imaging pipeline, and make sort of conscious choices about where those primaries will differentiate signals. So you can see in this example on the left from Hashimoto, they're trying to differentiate between two different types of crops that are in the same field. And the crops and weeds and soil and stuff have these spectral power distributions, which you can see are mostly the same throughout the visible spectrum, but differ at a few specific points. And so these points where they differ the most are assigned to the primaries, or the three primaries that they use um, to visualize things. And that results in having, part of the field be green and the other part pink, and allows you to differentiate, possibly at a distance, um, what exactly you're looking at, what are the boundaries between different things. And there's a lot of applications for these sort of techniques in remote sensing and things like that. As you can see on the right, another example of a blending technique. These are all aerial images for the most part, or different samples, geological samples or botanical samples, things like that. So um, there are multispectral functions in the world that are invisible to us, but um, if we could see them, it would allow us to have a finer discrimination or differentiation between different things. But the field of visualization shows us that blending colors, or selecting primaries and combining them over the same area, um, does not have as much information carrying capacity, as they call it, a word I particularly like, than taking those same primaries and expressing them in sort of a noise pattern like this. So this research group, Hauschinas and company, they conducted this experiment with sort of chloropleth map data, where um, they have all of these different demographic values that they're trying to describe for different states. And they sort of take an average of these primaries to produce these different colors for the different states, versus at two different scales, just populating the state with a particular primary at a particular frequency. So the spatial area that that primary covers will tell you how, like, the intensity of that particular data point. So this is kind of a preferred way to deal with multivariate data that's more than three variables with color visualizations. So we do something quite similar with the noise prism, where the intensity of each of these wavelength samples that we have is encoded with a related primary, and the frequency of that primary, the spatial area coverage of that primary in the resulting visualization, is sort of what tells us how intense that particular primary is. And the cool thing, in comparison to this visualization work where we're mapping things like home ownership rate to color, is here we're actually mapping color to color. And in the following slide, I'll explain how that works exactly. So we start with a spectral power distribution, a function of wavelength and intensity. But for us, we translate that directly to the number of pixels that should be used for each of these wavelengths. And the color of those pixels is described by this spectral sensitivity function, or color matching function, which is standard from the Commission of Illumination. And at each of these samples in this spectral power distribution, we have a particular tri-stimulus value, or three values which describe the unique color which is present there. And since each of these points are, really narrow band samples, as they would say, looking at sort of just a particular wavelength at a time, they tend to be extremely saturated. And so a process has to be conducted to convert them to displayable RGB values, which we then use to populate the prism. So this is a little demonstration of that here for this particular spectral power distribution, which has a very high weighting in blue, where you'll see the sort of connection to the prism is more obvious. It's not exactly the same because the prism, when it spreads light out into its component parts, the intensity is encoded in the intensity of that light, which is there, but each spectral part has more or less the same, occupies the same area. Instead, since we can't really play around as much with the intensity, we have limits to the intensity of the stimulus that we can display. We just encode that in the amount of spatial area that's taken up by a particular primary. So the first thing is to encode this spectral power distribution in the number of pixels, map it through the CIE 2-degree CMF to get RGB pixels to display. And these are the pixels that we have present in our pattern. Then we just shuffle that pattern so that it sort of covers the area, and we have a small process for mapping those colors so that now once they're integrated and once you look at the full pattern, it is fairly close in appearance to how that color would look if you had just been looking at the spectral power distribution of the stimulus itself. And what that allows us to do is sort of use these noisy patterns as overlays on actual color images so that you have some sort of higher dimensional information at each pixel. So here's another example of that. Now for a stimulus which is heavy in green energy or medium wavelength energy, not green energy in the traditional sense, but you know what I mean. And now you can see that this little prism spectrum has a much wider area in green. So that means there's a lot more green pixels present there. We shuffle that together, transform it for the display, and the appearance is quite similar to the integrated stimulus. And finally, another example for red. And you'll see that this doesn't really look red. We'll get into why that is on the next slide. But once we go through our little mapping process, it becomes red again. So if we had displays which could reproduce monochromatic light, or in other words, the most saturated colors that can be produced, then this would be a very simple method. It would be obvious and someone probably would have done it before. But since we don't have that, we have displays that have a limited color gamut. So they have particular primaries which sort of, outline the volume of this shape here. You can only reproduce colors that are within inside of this volume, whereas the sort of monochromatic primaries that we start with outline what's known as the spectral locus of light, which is basically the bounds of the most saturated colors you could produce physically. So the first thing that happens is what we call a gamut mapping technique, where we can scale down these primaries so that they fit inside of the RGB color solid. And then we have a simple mean mapping technique where we apply a nonlinearity or a power, which shifts the mean of each of these stimuli or these patterns to match the mean value of the stimulus itself, if it had been integrated colorimetrically. And you can see between these two Macbeth color checker charts, the result is quite similar in appearance, but we have some extra noise which tells us about their reflectance properties. So again, starting from the integrated stimuli, which are matching in color, but have different spectral power distributions, our technique allows for the differentiation of these stimuli. And the properties of these noise patterns tell us something about um, these spectral power distributions themselves. For instance, whether the spectral power distribution is narrow or if it's wider, if it has a lot of energy on the ends of the visible spectrum, and so on. And if you look at these at a smaller scale, you can see that they're sort of starting to integrate into the colors that they're supposed to look like. So it's an open question, the degree to which we can, the degree to which these patterns are legible. So what can we actually see that's different, or what can we see more than what normal color vision allows us if we use color distributions instead of solid colors. So there's been some research in this area. This research mostly comes from the field of texture synthesis, which basically aims to find what are the primary characteristics of texture that we can use to synthesize or analyze or describe textures. And so the initial experiments in this area, coming from Bell Labs in the 1960s, found that people can easily tell the difference between two distributions of brightness values or two distributions of black and white pixels by their mean and also by their variance. If the mean is kept the same or the average value, the average pixel value is kept the same, we can still tell the difference between two patterns if they have different variances. And later work, although it was refuted by this initial work, found that other distribution properties can also be perceptible, even if they have the same means and same variances. So what this shows us is that basically distributions of colors, distributions of brightness levels tell us more, they contain more information than just their integrated average values. So these experiments haven't really been done very extensively for color stimuli, though. So we had to do an actual psychophysical experiment to prove that people could tell the difference between two different stimuli that were metameric matches. And so the independent variables of our experiment were a bunch of color centers, so reflectance, Macbeth patch reflectance functions, and pairs of illumination metamers, so metameric illumination sources, which actually came from display primaries so that we could rescale them so that they match actually for each color. And this just basically provided us with a convenient stimuli set with which we could conduct this experiment. And the task was, given these four patterns, the observers had to find in a very short period of time which of the four of these is different from the other ones. So this case is pretty easy, but some of the cases were quite a bit harder. So just to review, the stimuli were generated in the following way. We have one reflectance function corresponding to this color, and we have two illumination sources which are sort of calibrated such that they produce SPDs or spectral power distributions which are equal in terms of their associated tristimulus values or the trichromatic visual perspective of them. And here's a little demonstration of the experiment itself. People are shown these groups of patches very quickly, and their task is to pick which of the four of them is different from the other one. So you guys can kind of get a sense for how difficult this experiment was. The idea was kind of to determine what's the threshold for color distribution differences in this context. So we wanted to make the stimuli hard. Maybe half of them people could see, half of them people couldn't see. That was kind of the goal. And the results showed that there's significant differences between the different color centers. and people's ability to discriminate between color distributions that have a different mean value. And also between these different illumination pairs, some of them were extremely easy to tell the difference between, whereas others were quite a bit harder. So all of these different display spectra have qualities that are a lot more similar to each other than they are to, let's say, the standard illuminant D65, which is very broadband, meaning that it has energy across the entire visible spectrum, whereas these other ones have uh, you know concentrated energy in certain regions of the visible spectrum. So we could at least tell that on average people can tell the difference between these different color distributions that we're using as visualizations for different colors, and also um, for different illumination properties. So this is an example that actually comes from RIT, from the Munsell Color Science Lab, where this is a computer graphics rendering of a bunch of cows, and they follow the Macbeth color checker chart patches. And the front half of the cow actually has different reflectance properties from the back half of the cow. But when displayed or when viewed under the D65 illuminant, the front half of the cow and the back half of the cow appear the same. You could see that the front half of this cow has this spectral power distribution, and the back is quite different. And in this visualization, I'm going to walk through each of the wavelengths in the visible spectrum, and the intensity of the cow's reflectance at that particular wavelength is going to be shown in this video. So as these guys are flashing back and forth, what this means is that at almost every wavelength, they have a completely different spectral response. But due to that trichromatic integration over the spectral sensitivity of a human observer, they appear the same nonetheless. So starting from this multispectral stimulus, we process it with our visualization technique, which now reveals the differences between some of these and others. It's not so clear. Again, we know that the spectral power distribution on the front half of the cow is not the same as the spectral power distribution on the back. And it's quite easy to see this, let's say, for less saturated colors and memory colors like skin tones, blue sky, foliage, etc. But sort of less common colors to see, like more saturated ones and things of the like, it's a lot harder to tell the difference between them. So that's an interesting property of this visualization technique. And here is a motion picture example of these multispectral visualizations being applied and sort of randomly seated at each frame. And this gives you kind of an idea of, I'm also adjusting the opacity as the video goes on. So at the beginning of the video, it's completely opaque, the visualization, and then in the middle, the visualization is completely at its peak. I don't know how well this is actually coming through now. I'm just thinking of this through Zoom. But the idea in showing this is basically that you can apply this technique over an image or a movie, and since filmmakers like to have grain in their images, it could be sort of a convenient way um, to both increase this aesthetic quality, cover up artifacts, and also include additional information about what's actually being seen, and or basically differentiate between different colors, or add sort of a living depth or sort of motion to different brightness and color regions that, for instance, film grain is known to do. So the grain characteristics of film behave differently for different brightness values and different colors, and this kind of allows for the separation of different parts of the image in a sort of more visually engaging way. And this has actually been implemented in FilmLight's Baselight color correction system. they plan to release it in the with their next release of the software, so that artists can actually make use of this type of technique um, to beautify their images and perhaps, I don't know, communicate something more about what's present in the scene. Um, so moving forward, we're doing a collaboration with Jan and Peter Moravik from HP. They work in printing, so they work with different types of pattern arrangements for halftoning and things of the like. So a big part of this experiment and this work was determining a way to sort of predict how perceptually different these visualizations would be to an observer. And an obvious factor in that is the actual pattern that's present. So how big each of the pixels are and how they're arranged. So these new experiments kind of focus on two different types of pattern arrangement. One where common colors are dispersed from each other, which sort of lends to the stimulus being more easily integrated, and one where similar colors are sort of maximally clustered together. Um, so common colors kind of form these bigger dots, which one could imagine might make it easier to interpret these patterns and see what are the component parts. Whereas, these patterns might make it easier to blend the patterns in um, with the image behind it. And the results were quite interesting. They showed a similar trend in a difference of sensitivity for different color centers, and a similar trend for differences in the illumination pairs. But there was a significant difference between these two types of patterns, these very clustered patterns, which they call green noise, and these more dispersed patterns, which they call blue noise. And we found there was a significant difference in those for some colors, but not other ones. And the point of this work is to kind of test various color difference metrics and texture difference metrics to see how well they can predict this ranking and this sort of visual phenomenon that we've measured. And that's what we'll be presenting at the Color Imaging Conference this year. So hopefully I'll see some of you there. And thanks a lot for listening to my talk. I'll now open the floor for questions and comments and all that stuff. So thanks a lot.
Thank you so much, Trevor. We will give you a hand as much as we can online, but very, very interesting work.
Thanks.
And yeah, great presentation. Thank you so much. Maybe stop sharing your screen and then we'll see if anyone wants to turn their camera on. You're welcome to unmute and just ask a question. Maybe I can start the questions.
Okay.
I'm wondering if this technique would be helpful for people with color vision deficiencies at all. Could it help? Have you considered that?
I actually have not considered that, but I think that it could, because it is all about differentiating between colors which appear similar to people. It's certainly not optimized for that in particular, but yeah, like the extension of the visualization to a distribution of colors sort of calls upon other visual cues that they might be sensitive to, um, that yeah that they would assist them in differentiating between colors. But like I said, as you saw in some of the visualizations, it does sort of blur spatial boundaries and stuff like that. So that might actually be more annoying to them than not. But for a particular, uh, I guess, color differentiation task that's sort of contained, then yeah, it could be very helpful. Thanks for that comment.
Hi, I'm a little confused as to the application. So you're making it a nice a nice way to visualize things. But what about just computing the metamer mismatching? Wouldn't I like to have a quantitative thing using the algorithms that exist?
Sure. Yeah, you might want to quantify it, and there, there's different ways to sort of redistribute primaries within, the conventional imaging ecosystem to um, sort of show those differences between things. But yeah, the application generally is to sort of explore what extra information can be conveyed in color distributions versus um, independent colors on their own, and to look into that a bit more.
Okay, so I saw that with the Medi-Cal example. But, there you have a uniform or two reflectances. In a real image, you'd have too much texture to deal with, I assume, to see the difference. Like if I were you know what I mean? I mean, that's a very synthetic example. And if I took a picture of, I don't know, the bookshelf behind me, you wouldn't be able to tell
Sure.
yeah.
So I've actually used some of your multispectral samples, some of the CG scenes of different rooms and things like that. And one possible application I found was to sort of differentiate between. Um, ooh, I don't know if I'm frozen here.
No, you're okay.
Oops, sorry about that. Okay. So, for instance, in a multi-illuminant scene, you could have two illumination sources which are illuminating, um, let's say, the floor of the room, and the reflectance of the floor of the room is metameric for those two illumination sources. Um, and this visualization technique would actually show you the boundary of where those two illumination sources are present. So this could possibly be useful for architectural visualization, something like that. Even in your examples, which had not metameric illumination sources, but two different colored illumination sources, the results did show clearer boundaries in terms of where those illumination and shadow boundaries are. So yeah, the work on the application is still underway. The next experiments we want to do are kind of visual search tasks that are more application relevant. Um, but yeah, it's not only just real world applications of seeing um, if we can see more details or differentiate between more real world things, but we can also do synthetic examples where we have multivariate data like those visualization, the color weaving and color blending things I showed earlier. Um, so yeah, that's the idea. See if we can have sort of a higher dimensional visualization using color.
Okay, thank you. And I look
Yep.
forward to talking to you in Montreal.
Okay. Yep. Likewise.
Is there any application for this in encrypting the images?
Yeah, there could be. Yeah, steganography, I believe it's called, where you
Yeah.
you want to sort of hide information in images.
Mm-hmm.
Could be, sure, but we haven't really looked into much like analyzing the images themselves. So you could encode it inside of the image, but then there's the sort of process of decoding that information back out. But yeah, you could, have sort of different noise characteristics in certain areas that if they could be targeted and analyzed, then um, it could be useful for that. But of course, that's hypothetical.
Yeah.
Thanks for your comment.
Yep.
I'm wondering if there's other sort of data sets that could be applied to. I'm just thinking like maybe astronomical
Mm-hmm.
images where it's difficult to pull out features might add
Yeah,
to ways of interpreting data.
astronomical images could be really useful and kind of might lend themselves well to this technique because we have, in that case, various like point source stimuli that have open space around them where the stimulus could be spread out into a noise prism visualization that could give people an instant clue sort of as to which spectral bands are most intense at that location.
Any other questions or comments or questions about Trevor's work in the film industry, which I would love to hear a little bit about. What did you work on any particular films or any?
I don't have any question because I think it goes beyond what I can understand completely. But I just want to congratulate your work because I think during my doctoral project, that is something that they asked me about how the combination of like what is the final spectrum of the combination of light and the color that we applied in surfaces, and I wasn't able to respond that because I think it goes beyond of what I can understand, but I think it's a very interesting work, and sure, I would like to know a little bit more about the applications of that in architecture, in visualization, or VR, etc.
Sure, yeah. So the work is ongoing, and hopefully you'll see more of that stuff. Yeah, it goes beyond sort of the area that people generally think about, and also beyond what most of the color community would consider as particularly useful. But I don't know. It sort of touches a certain philosophical interest in me, and I think there's definitely room for useful things to come of it. At least I hope. But thank you for your comment. Appreciate it. Robin, to answer your question, sort of the biggest project I worked on was an independent film from a Spanish filmmaker called Aaron Siebert, and it was about the sort of struggles of the maternal line, like his maternal line in Galicia, which is traditionally a very poor province in Spain. And we had we did the process during COVID, so it was all online. So all over Zoom, and a lot of color management problems and stuff like that, and miscommunications in terms of color terminology. But it was sort of a intimate process working with the director over the course of a year, constantly revising the color rendition of the film. The big problem was I kept on rendering scenes. I was living in Barcelona at the time, so I kept on rendering them sort of with the ecology of Barcelona in mind, which is quite different from Galicia, which is a lot more saturated and green, and there's a lot more rain that happens there. And so that was sort of an interesting bit. But the film was submitted to a number of festivals around Spain, some of which were sort of closer to me than they were to the director. So I had the opportunity to show up at those festivals and receive awards and give award speeches in Spanish, which was fun. I really enjoyed that. Otherwise, I've worked on a few music videos recently. I have a lot of musician friends in my hometown, and we work together quite frequently. And other than that, film school, a lot of my own personal, like science communication sort of projects. But the internship at Company 3, New York, was kind of a big step for me in 2016, where I actually got to see sort of how technically a professional post-production house works. And they were working on sort of big projects like HBO's Mr. Robot at the time, and there was a movie called Gifted we were working on at the time that came out in 2017. There was some remake of Cinderella with live action characters, Emma Watson. Company 3 is like basically the biggest color correction house in Hollywood. They do all the Star Wars and Marvel stuff, and so it was very interesting to see kind of how that industry works. And since I had that opportunity, that's where almost everything else came from after that, working in the image processing for enhanced cinematography lab. And before that, I worked in another post-production house in Rochester, who basically hired me to tell them how a professional post-production house works and how they should outfit their equipment and all that sort of stuff. And yeah, so the work I did in Spain was the first project I was sort of brought on for was this high dynamic range for the European Union project, where all of the different types of film equipment manufacturers, from display manufacturers, projector manufacturers, camera manufacturers, um, color correction and image editing software people, and also different production professionals. We're all involved to sort of collaborate on how HDR technology should be used, and yeah, how to make everything work together. And so, yeah, I've always had a sort of affinity for the motion picture world, and the fact mostly that they'll go to great lengths to have sort of perfect images, or very, very high quality images. They're willing to do very tedious and expensive operations to make that happen. Um, Sometimes in spite of their resources and the interest of their customer base and everything, but they really just care a lot about reproducing what they see in the world. So um, that's quite important to me.
There was a comment in the chat by Sarah. Sarah, do you want to just ask that, or are you?
Sure. It's right there in the question.
Yeah.
You mentioned a minute ago that there's something about this project that piques your sort of, you described as your philosophic interest. Could you talk a little bit more about that?
So yeah, it's sort of peeking into, I guess, what are the limits of color vision? How those limits are informed by stimuli that are present in the world, sort of what could our perceptual experience of the world be if we did have a higher degree of color discrimination capability. And yeah, I guess I'm sort of putting that under the philosophical term. I don't know if that's the appropriate description for that, but from what I've seen from color philosophers, they kind of look into, what is a color? What does a color stimulus mean? What's the value of that, and so on. So yeah, that's basically the sum of it.
I like I said in my first comment, well beyond my field knowledge in every single way, but I was really interested in the cow, the Macbeth cow, whatever it's called, the cow. As it ran, I realized that the cows that were lower in value, very surprisingly to me, the front half of the cow, as it started to disintegrate, looked darker than the back half of the cow. It's the reverse for the lighter cows. And I assumed, and this may well be entirely wrong, that this is a perceptual experience, that the value contrast between the light part of the reflected light, the bright, the bit of the cow that's reflecting light, in contrast with the sort of color of the cow, it's that contrast that is making the cow appear darker. It's
Sure.
a Bezold effect thing going
So,
on.
I don't think it's a illusion. It could come from a few different places, because the goal of the visualization is just to have two different spectral power distributions on the front and the back half of the cow, which are rendered the same under a particular illumination. But those spectral power distributions can vary in any number of ways. And the result of those spectral power distributions, when rendered through our visualization technique, which has sort of its own interpretation that's imparted, may also make something look brighter or darker than something in comparison. For example, the best way that I know or that I could think of to render spectral power that's at the edges of the visible spectrum is with a very low intensity, because I'm mapping basically directly from how sensitive we are to that particular wavelength to the visualization. So what tends to happen is if you have sort of this like spectral power, you could have two different types of spectral power distributions. One that cuts off before getting to the edges of the visible spectrum, and one that sort of reaches the edges of the visible spectrum but still has energy continuing outwards towards that. And the result of that is you have a bunch of black pixels if you have a high intensity at the edges of the visible spectrum. And there was some debate about that actually at the Color and Imaging Conference, if that's the best way to interpret that. I don't know how useful it is to show that there's high energy towards the ends of the visible spectrum, but that is something that the visualization technique is good at doing as it's formulated currently.
Yeah, thank you.
But there could be also a perceptual effect, but I would guess it's probably more from the first two things, the actual stimulus itself and the visualization.
Yeah.
There may be an illusion as well to be gleaned from that.
Yeah. Thank you.
Yeah, thanks for your question.
Thank you so much, Trevor, and thank everyone for their questions. Just one more comment. I think, Sarah, with this, technique for discriminating things that appear the same, it kind of adds to our way of knowing, and it might actually add some further dimension to how we, understand the world. So I think, if you ran some art images, it would be really interesting to see there. Sure. Or you might get, with restoration, or if a painting has been restored, what does that look like? What do the areas look like? So that might help discriminate that. But I think it's really interesting to see, and I'm happy to have like such a strong kind of scientific component that is then going to spill into other visual components. So this sort of color is where everything meets, right? So we've got art and science and philosophy, and I think that's why, and architecture and space, and your comment about how the color of a locale was very important because it helps define, it defines the spaces that we're in and the places that we go. So colors is very beautiful, all-encompassing world for so many of us. So thank you so much for just adding a whole new dimension to us, and very happy, and congratulations once again. And thank everyone for coming. Our next speaker is on color vision deficiencies, Jeff Hovis from University of Waterloo. So check our website. I think that's October 24th. And then we have Doreen Balabanoff speaking about color in architectural spaces in the birth environment, which is at the end of November. So lots of interesting things to come. And thank you again so much, Trevor.
Awesome. Thanks a lot. Appreciate it. Thanks all for coming and for your awesome questions and comments.
All right. Good night,
and for
everybody.
With the award, I appreciate it.
Very happy to make that possible. All right.
Good night.
Bye-bye.
Bye-bye.
Bye.